Okay. So before we get started, Ruchika, Dr. Prakash asked me to make a few announcements for CCBBI. She's going to be like a minute or two late. But first off, so please welcome Adam Sharp, our new office assistant at CCBBI. He's going to be helping us with all things that are center related. Adam, do you want to wave so we can all see you? Perfect. Hi.
Yeah, thank you for the greeting. It's nice to be here. I went to undergrad in Miami, Ohio, and I went to the East Coast for grad school. And I'm back here in Columbus for work, and I'm glad to be here.
We're glad to have you. Thanks for joining us. And then, so second, CCBBI is hosting a webinar on Tuesday. And we're going to give an overview of the types of research that we do at CCBI and discuss diversity and other research initiatives. And I'm going to post the link in the chat so you guys will be able to click it and register for it. All right, so let's get started. So it's my great pleasure to introduce Dr. Margaret Sheridan today. So Dr. Sheridan is faculty at UNC, and she got her PhD in clinical psych from UC Berkeley. And after her clinical internship, she was a Robert Wood Johnson Health and Society Scholar, and she postdoced at the Harvard School of Public Health, where she worked with Dr. Chuck Nelson and at Harvard and John Gabrielli at MIT, which is how I met her. And then she worked as an assistant professor at Harvard Med School for a few years, then left for UNC in 2015. And so there she's the director of the Circle Lab. So Margaret has an amazing grant and publication record that I'm not going to get into. She's a whopping H index of 44, and it's pretty clear why, right? So she does incredibly important work and translational work where she investigates childhood poverty and other adversity as it relates to neural development. And she approaches this very big and important question in innovative ways. And I was fortunate enough to get to know Margaret when I was a grad student in the Gabrielli lab. We bonded over shared interest in child development, and we collaborated on a few successful and unsuccessful projects. And we started up a journal club where we geeked out over the basal ganglia, and she got us these wonderful mugs to commemorate, which I still have. And she's, Margaret is thoughtful and supportive, and she's not at all afraid to cross disciplines, techniques, neural systems, whatever is needed to answer her questions. And she's also a wonderful friend and collaborator, and I'm very lucky to get to know her. So please join me in welcoming Dr. Sheridan.
Well, that was so lovely, Zainab. It is such a pleasure to be here. It's the most fun thing about getting to know people in your career is then getting to continue to interact with them across the years. It's like just the funnest to be able to come and give a talk where Zainab is a professor, and it's really lovely. So thank you. I'm going to talk today about the dimensional model of adversity in psychopathology. And this is the model is a model that I published in 2014, and I'm going to kind of describe what it is and why I study it. And at the very end, if I have time, I'm going to try and make time. I'm going to talk about a couple of new projects, and I would love to hear everybody's thoughts. Most of what I'm going to talk about today in terms of the neuroimaging is going to be structural data. I also do functional MRI, but it just hasn't made it into this particular work quite yet. But that is kind of the next step. And so talking about those projects, I'm excited to get people's feedbacks and thoughts and about that stuff. So I study, I'm always a little confused about this. The main question which drives all of my research is how is brain development linked with adversity, and how does this contribute to risk for psychopathology? That's kind of the fundamental underlying question I'm trying to answer. And the reason why I'm trying to answer that question is because child adversity, that is adversity experienced, so adversity experienced during childhood is associated with long-term deficits in health and well-being. And we know that through randomized trials that this association is causal, so that adversity experienced during childhood causes changes in health and well-being later. And as a result, I think it's less useful and important to just continue to document this association with more research. But instead, I think it's important to ask the question, how does adversity get under the skin to affect health and well-being? So what are the models that we can use to understand the mechanistic relationship between adversity experienced early and outcomes in childhood and adulthood? And one very common model is a cumulative risk model, and the ACEs is kind of part of that model. And in the cumulative risk model, the conceptual idea is that early in childhood, exposure to adversity accumulates. So all different kinds of adversity are, if you have one of them, it's bad. If you have two, it's worse. And they sort of accumulate to increase the stress that you're experiencing. And then that stress leads to changes in your body and downstream consequences. And there's a lot of great things about the cumulative risk model. One of the great things is that it's a great model for predicting who is going to have psychopathology. If you're a clinician and you're sitting in your office and someone's in front of you and you need to decide who needs the treatment the most, the question you should be asking is how many adversities they've experienced, because that's a very good way of figuring out who's going to have the most psychopathology or the most health problems. It's not a great mechanistic model. And one of the primary problems I have with it is that it really isn't asking the question from a developmental stance. So we know from developmental psychology and neuroscience that there are very clear ways that experiences during childhood are embedded into the brain. So your experience with vision shapes your visual cortex, your experience with audition shapes your auditory cortex. It sort of makes obvious sense that these kinds of adverse experiences would also shape your brain using those same developmental plasticity mechanisms. And so the dimensional model really fell out of that observation and kind of consternation with the world of adversity research, that it wasn't kind of taking into account the fundamental and important idea. And what I and my colleague Kate McLaughlin ended up doing was thinking about the kinds of experiences that were housed under the name adversity, and thinking about the ones that had the most evidence associated with them and whether or not they could be organized into kind of dimensions of exposure. So we ended up proposing these two dimensions of exposure: deprivation and threat. And by threat, what we meant were exposures to things like physical and sexual abuse, domestic violence, and community violence, so interpersonal violence exposures. But what would go into this exposure is definitely other things like exposure to natural disasters or to war and terrorism, things where there's a clear threat to your life or the life of a close other, and that threat is kind of fairly unpredictable and clearly quite dangerous. By deprivation, what we were thinking of is, instead of the presence of this salient learning experience in childhood, that's how we were thinking about threat, it would be the absence of expected learning experiences. So species expected experiences that parts of your brain were sort of prepared for receiving. So here we began this research by thinking a lot about institutionalization, since that was some research that I had been doing when we were developing this theory. And I'll talk about that a little later in the talk. But we were then thinking that institutionalization, that experience of that profound deprivation of a caregiver, would be similar but worse than something like neglect, or the kinds of exposures that can go along with poverty, where the primary caregiver for a child, or even many of the invested caregivers for a child, are so under-resourced that they're unable to provide the kind of scaffolded cognitive and social stimulation that the child's brain is prepared to experience. And the kinds of experiences we were thinking of here when we talked about deprivation were really like the scaffolded learning experiences that kids, babies and toddlers and young children usually get as a part of their caregiving relationship. So less the kind of warmth and attachment, and more sort of what those actual experiences are, what the kids' brains would be expecting to receive. So some basic principles and limitations before we get into this. One is that we are not trying to say that these two dimensions describe every piece of childhood adversity. For example, there's a lot of current work on unpredictability. That's another potential dimension or another set of experiences that kids have. We're not trying to describe everything. We're describing two dimensions that we thought there was a lot of existing evidence for. Also, we're really talking about the proximal inputs into the child's developing brain, and we think that those are going to lead to proximal outcomes. So by this, I don't mean the intention of the parent or the degree to which the parent loves the child, or the sort of conceptual understanding that the parent or the kid has of what's going on, but just the actual experience that the brain is receiving. So we really wanted to move away from what had existed in a lot of the adversity literature, which is sort of a lot of description and concentration on sort of the positive nature of the caregiving relationship, and really just talk about what is the brain, what is happening to the young child's brain, and how can we think about those inputs shaping brain development. The same way that you wouldn't ask about whether or not somebody wanted to have visual, like the parent wanted to provide the child with visual input if the child had, say, occlusion of one eye, you wouldn't say, did the mom want to give the kid input? You would just ask, did the eye get input? That's the same kind of thing we were trying to do here. And then importantly, we think that these are going to lead to proximal outcomes. So instead of thinking about the long-term health and well-being, the psychopathology is the final outcome, we were actually asking what mediates that relationship? What are the pathways that you get from adversity to psychopathology? So it's that pathway piece that we're going to try and delineate. So we had some hypothesized pathways for threat and deprivation based on existing animal literature and work in humans. And our idea was that threat experience was an early learning experience that was these enriched for violence, and they were going to thus enhance neural systems which are going to support dealing with danger. So if you take an example from the mouse literature or the rodent literature, I apologize because I technically don't know if they are rodents or mice, or they are mice or rats, but they are rodents. In early puphood, there's this normal attachment learning. You have a normal relationship with the pup, and it licks and grooms you, and it's got appropriate bedding, so it's not stepping on you and sort of doing abusive behaviors. What you see is that in early infancy, they basically infant pups will sort of learn in an attachment way, so they'll move towards any form of stimulus at all if it's associated with mother. And then in later infancy, if the mom is present, you continue to see that kind of like attachment style learning where they kind of affiliate with many different kinds of stimuli. But by the time you get to late infancy, you see a fear aversion evolving where they move away from stimuli which are negative like shocks, and they move towards more positive stimuli like food. If you take pups and instead raise them with a mother who is abusive, who sort of does really negative and caregiving behaviors, throws pups around, doesn't lick and groom them, or if you just shock them when they're in early infancy, what you see is that in early infancy, you still get this attachment kind of learning, but here in later infancy with the mom, you now transition to having fear aversion. So you get an earlier acquisition of fear learning, which makes sense if you're going to be born into an environment in which that lots of dangerous things could happen to you. One thing would be to figure out when they're happening and why. This is work by Regina Sullivan. In humans, we also observe the same enhanced early fear conditioning in childhood. So one of the things we went into the theory asking about was fear conditioning, because we thought that was something that very logically should go along with exposure to threat. And when I got to UNC, I was able to test this idea in a small study. So this is just 64 kids. And we brought this group of kids in between the ages of 4 to 7 years. We measured deprivation and threat. So here we're defining deprivation by the child's report of their neglect, the parent's report of their cognitive stimulation in the home, and parent education. That's a variable that we have not used going forward, but at this time we were still using parental education as a proxy for deprivation exposure. And for threat, we asked the child about their own exposure to violence. We asked their parent to report on domestic violence. And then there's a questionnaire which sort of indicates risk for abuse that we included. We controlled our analyses for age, gender, and IQ. And the form that these analyses, well, I'll tell about that in a minute. So here this was a pilot study. All we were able to do here is just behavioral tasks, but we did one behavioral task, which is a cognitive control task. This is a Simon task where we either show the child a pink dot and we tell them to press on the same side of the screen, or we show them a yellow dot and then they press on the opposite side of the screen. Very simple task. They just do blocks of congruent and then blocks of incongruent stimuli. And this requires cognitive control, because when you're doing opposite side, you have to withhold your response to the same side and instead press with the opposite hand. We also had them do a fear learning task. And this was a task that it's used with very young children because it sort of facilitates fear acquisition in a really short period of time. So we showed them blue squares, and nothing happened when we showed them blue squares. And then we showed them blocks of orange diamonds. And some of the times when the blocks of orange diamonds were shown, they heard an adversive stimulus. So it was like a loud kind of screeching sound that's been used previously in fear conditioning with little kids. Sometimes they saw blocks of the orange diamond and nothing happened. And what we were measuring were two different things. One was skin conductance response to the sound, the like adversive stimulus, and one was skin conductance to the orange diamond when there was no sound present. So the second thing is true fear learning. Have you acquired this association between the sound and the stimulus, even when the sound is no longer present? And the first thing is really just your reactivity to negative stimuli. And what we found, the way we do these analyses in general, and this will be what I describe going forward when I talk about deprivation and threat analyses, and I am super happy to talk with people about this, is we're just running a regression. So our question isn't, are there kids who just have deprivation, or are there kids who just have threat? But instead, most of the time, these adversities co-occur at the level of the population. And so what we're trying to ask is, if you look at the amount of deprivation that you have, that's going to vary, and you control for the amount of threat that they have, which is going to vary. Do you see a relatively stronger prediction for deprivation versus threat, or threat versus deprivation, depending on the variable? And here our predictions were very consistent with our hypotheses. We found that the amount of deprivation that the child had was related to their performance on the cognitive control task, whereas their deprivation went up, their inverse efficiency went up. So the degree to which they were slowed and less accurate on the task increased. So they did less well. And we similarly saw that the degree to which kids responded in terms of their skin conductance to the unconditioned stimulus, that loud sound, was related to their threat exposure, controlling for deprivation. And that is that as their threat increased, they had a more blunted response to the unconditioned stimulus, which is this kind of blunting following threat exposure has been replicated a number of times. So that kind of blunting was the expected response. Most interestingly for us, but this is a very small study that we're trying to replicate now, was that we actually found earlier fear learning. So that we found that kids who were in the high threat group, those are the red dots, differentiated between the CS plus and the CS minus more than kids who were in at earlier ages than kids in the low threat group. So the slope of the red line is significant, and the difference in that younger ages is also significant. Obviously, it's a tiny little sample, and we're trying to follow this up now with more data collection that I'll tell you about at the end. So that's one example of a study that we did at UNC. We've actually done this a lot. So we also found in a separate study reduced differentiation between fear and safety cues in adolescence. So we see this early acquisition and then reduced differentiation later. And there are some theories about the development of stress sensitivity that are kind of consistent with that idea. We also just see globally increased reactivity to negative stimuli for threat controlling for deprivation, and poor emotion regulation for threat controlling for deprivation, which is kind of consistent with this, consistent with this idea generally that we expect threat to shape the neural systems involved in fear learning and salience processing, including the amygdala, hippocampus, and medial prefrontal cortex. And it's going to shape those systems to be more reactive and to take longer to sort of come down from that reactivity. And we've done this work both in terms of observing it in terms of bold activation differences for kids with and without threat exposure, controlling for deprivation, behaviorally, like I just described, and in terms of self-report or sort of experimental manipulations of emotion regulation. In terms of deprivation, as I described, this is really different than the presence of a learning experience. This is kind of the absence of a learning experience. And the way that I first was conceptualizing this was very similar to sensory deprivation. So I didn't have a particular reason for thinking, about literally why. So often people often ask sort of why would deprivation do this to the brain? And I guess I wasn't really thinking about why deprivation would do this. I was more thinking of a theory by analogy, which is that we know if we deprive the brain of sensory information, it develops in a particular way. And one of the things that happens is that the number of synapses, if you just deprive all visual input into a kitten's eyes, what you see is a reduction in synapses. And if you deprive humans of visual information, you see a reduction in cortical thickness. So I was really just saying, like, in my head, I was thinking, well, that seems like what we should see is a reduction those same kinds of reductions, because I'm thinking of a reduction in an input that I imagine would be associated with association cortex, the kind of things that would shape prefrontal and parietal development. And I was further thought that this was the right way to be thinking about it, because I knew that really gross deprivation, so being raised in a cage as a rodent versus being raised in a cage with like fun things to play with or being raised outside, resulted in decreases in cortical thickness and volume, really generally all over the brain. And I had recently seen that same finding in kids who'd been exposed to institutionalization. So this very global deprivation negatively impacted global volume and thickness in middle childhood. And the way we were imagining this working was sort of in early childhood and toddlerhood, what you would have is you'd have this kind of scaffolded caregiver interaction around cognitive development and social development that would lead to a rich dendritic arbor, because it would increase all the firing of all of those different synapses, and that would lead to lots of dendritic spines and branches. And we would measure that in the brain as cortical thickness, so increased thickness. And that, in contrast, kids who were exposed to deprivation would have less of those caregiver interactions that would result in reduced dendritic branching and then cortical thinning. So the idea here is that deprivation is the lack of species expected input. We did observe this cortical thinning and reduced volume in middle childhood. And recently we followed those kids up when they're age 16. And I'm just going to describe what we've observed in that most recent data, which is kind of interestingly consistent, somewhat not with our original hypotheses, which is sometimes how hypotheses go. So the Bucharest Early Intervention Project is a randomized a project with a randomized assignment from kids who were living in institutions in Romania in 2000 into foster care intervention. So there's 136 kids between 6 and 30 months living in institutions in Bucharest and 72 community controls. Sorry, this is a study that was started by Chuck Nelson, Nathan Fox, and Charlie Zena. So not me, obviously, I came late. But I was able to analyze some of their data. So in these institutions, they were separated from their parents, the kids. There's these primary impacts that primary results of being separated from your parents, which is that you have the lack of an invested caregiver. So that means that there were few responses to distress. There was very little cognitive and social stimulation. There's a very regimented daily schedule, so not a lot of variety and changes in the kid's schedule. And there was very non-individualized care. So if there were inputs into the child's cognitive and social inputs, they weren't really individualized to the child, which is not the way that human brains are used to developing. There's also this sort of lack of psychological investment by caregivers. So they have rotating shifts and really high child caregiver ratios, which means that the caregivers don't get a chance to really form a strong relationship with the kids. And then there's a secondary vulnerability to exposures like traumatic violence. So we're really focusing on institutionalization as this lack of an invested caregiver, this sort of like lack of overall input to the child's development. But it's useful to remember that those other two pieces are also here. And as these kids have gotten older, we've begun to be able to ask them about their other kinds of exposures, like to traumatic violence. Although at this point, we don't know about any of that with the kids. They were screened in a lot of other ways, which I can talk about if anyone's got some questions. So there's this baseline assessment, just to describe how this works, where they're brought in, they exclude some kids there, and they are excluding them based on whether or not it appears that they have a genetic problem or if they have been exposed to alcohol prenatally. So that's how they're sort of initially excluding kids. They take half of them and they put them into foster care, and then those kids are followed up at 5 years, 8 years, 12 years, and 16 years. And there's an MRI here at 9 years and then at 16 years. And then similarly, there's a care as usual kid group. That group was not randomized into foster care intervention. They stay in institution and they're moved in and out of the institution as like the sort of development of the foster care system in Romania. And again, they're following the same time points. At 9 and 16 years, we're able to measure their neural structure. And at 16 years, we get a slightly larger sample than we had at 9. So we have 115 kids. Their data was acquired on a 3T Siemens with a 16-channel coil. There's 41 kids in the care as usual and foster care group and 33 in the never institutionalized group. And of those, there are 64 children who also have data at time 1. And so there's a slightly smaller group of kids, obviously, that have all of their data at time 1 and time 2. And the NIG group is particularly small. I'm going to report on some findings in that group anyway, because it's the sort of like longitudinal change is quite important. We also image them on 2 different scanners. They have the same field strength and the same maker. They're both by Siemens, but they were on different scanners and they're using slightly different pulse sequences. So the longitudinal data has some problems. We've tried to address them by looking at heteroscedasticity, and it does seem like the variance is the same in both groups. And so there's a good arguments to be made that you can do the analysis, but I want to be upfront about that as we look at this data. So if you just look at the overall decreases in cortical thickness from age 9 to 16 years. This is one of my first questions is like, does that look normal given that we've got them on different scanners with different pulse sequences? It does look normal. We also looked at increase in white matter. It does increase between 9 and 16 years. The sort of areas where we're seeing more decrease make sense in sort of the whole lateral and medial brain, but less in say the motor strip. But and we're looking at all of our whole brain results here are cluster corrected from multiple comparisons. And that's important to know because at age 16, if you just look at care as usual versus foster care, what you actually see now is thicker prefrontal cortex, and it's specifically in the anterior cingulate and the IFG for kids in the care as usual group versus the foster care group. So previously, we had never seen any differences based on random assignment before when looking at this data. So it was first very powerful to observe this. This is one of those pieces of evidence which is causal. So we know that the difference between the care as usual group and the foster care group is due to their exposure because they were randomly assigned. So that, first of all, is very powerful. It was also really striking that it was in the opposite direction as I expected. So we were actually seeing thicker cortex for the deprived group. And then if you look at the ever institutionalized versus the never institutionalized, you again see that. So we see thicker cortex in some very similar areas for the kids in the ever institutionalized group versus the never institutionalized group. Now, when I first saw this data, I was pretty freaked out. And I will say I was very heartened because another group published a paper. There's a group of Romanian orphans who've been studied in England. It's called the English Romania Adoptee Study. And happily, they found the same thing, so thicker cortex, in the same areas. So they also found it in the IFG, the anterior cingulate, and the temporal pole. So I felt better that we weren't the only people observing this and that it was maybe real life, but it was quite surprising. And the thing that I ended up doing is looking at what was happening between 9 and 16 years to sort of answer this question, why are we suddenly seeing different results? And the answer was quite interesting, which is that foster care kids thin more across time, and so do ever institutionalized kids. And the areas where they thin are similarly in the prefrontal cortex. They don't perfectly overlap in this whole brain analysis, but that is, we were seeing lots of midline regions thinning and then also lateral inferior frontal gyrus thinning for the kids in the ever institutionalized group versus the never institutionalized group. So those never institutionalized kids are following the typical developmental trajectory from 9 to 16 years in terms of thinning, and they're thinning more than the kids in the ever institutionalized group, and similarly for the foster care kids. So we see this sort of thinning and reduced volume in middle childhood, but it seems like we also see this change in developmental trajectory that was unexpected. Now, we've also looked at a bunch of other ways of measuring deprivation, neglect, measuring in the home, what the actual cognitive stimulation is like. And we've observed across a bunch of different samples here that you do see this impact of a lack of cognitive scaffolding in the family, and a lack of social and cognitive stimulation is associated with deficits in executive function and language. when you're controlling for exposure to threat. And I should say that in these situations when we're controlling for exposure to threat, it's not like mild threat, it's pretty severe threat. Most of these samples are selected to be enriched for exposure to abuse. So we're not comparing sort of, this is always to me the more surprising finding, that there's anything that's more strongly related to deprivation, because most of the time we're actually not looking at institutionalization, we're looking at something more mild than that, and we're comparing it to frank abuse that would be reportable to Child Protective Services, and sometimes is. So I think from this work, it seems to me that there's pretty good evidence that deprivation sort of broadly shapes the development of association cortex involved in higher order cognition, including the prefrontal and parietal cortex. And I would say that like at this point in theory development, I was feeling pretty positive about the data as it was lining up. I think there's still some holes and things that we want to spend some more time answering, lots of them. But it did sort of feel like that initial theory was developed based on like animal work and a little bit of existing literature, and this was feeling like it was supporting the theory. But there's a lot of leftover, I think that's not what I want to ask about yet, sorry. So there was a lot of leftover questions. One, of course, is whether or not this actually leads to psychopathology. So it's one thing to say that we're seeing this relationship with the mediator, but we don't really have evidence that that's on the pathway to psychopathology. And so we've been able to ask this question in two different projects in a more limited way. This was work done with Adam Miller in my lab, or he's no longer in my lab, he's actually a research assistant professor now, but he was in my lab. And we use data, done two existing data sets that are longitudinal in nature where we can really ask this mediation question. And we've used structural equation modeling. We don't have any brain data yet to serve as our mediator, but we've been able to ask the questions using non-brain outcomes. So this child development project was collected a long time ago. It's about 585 kids. And when the kids were age 5 and 6, they went into the home and they did an observation. They asked about the number of toys and books that the child had, the number of books the parents had, whether or not there was an appropriate play area for the kids to play in. And they also asked the parents about the child's exposure to abuse from the time they were 0 until 5, and then the parents' own use of harsh discipline and the existence of family conflict. And then they observed any hostility or rough physical contact during the actual interview. At age 14, they measured the child's vocabulary, and at age 17, they measured their internalizing and externalizing symptoms on the CBCL. And what we found is that deprivation mediated the association between deprivation and externalizing psychopathology was mediated through the verbal abilities, that vocabulary at age 14, and that this mediation pathway was significant. And in contrast, threat exposure had a strong direct relationship with externalizing and internalizing psychopathology, but it was not mediated by verbal abilities. Now, in this project, there just wasn't a measure of emotional regulation or functioning outside of psychopathology that we could really use as the mediator, so we weren't able to test that alternate pathway. We were able to control for socioeconomic status, so we were able to sort of say this is net of general risks associated with socioeconomic status that are related to psychopathology. And then we recently replicated this in a paper using the Fragile Families data set, so it's a slightly larger sample. Again, it has this longitudinal nature. Here we're looking at earlier deprivation, earlier verbal abilities, and earlier psychopathology, and we're basically getting the same finding. We're seeing now verbal ability also mediates internalizing psychopathology, so both of those. But we, again, see this kind of direct effect of threat on internalizing and externalizing psychopathology, and this indirect effect through verbal abilities for deprivation. So sort of consistent with this, the theory, it kind of laying out the whole model. The final question that I had that I thought could be answered with existing data before trying to get any of my own, well, you know, also one needs time to write grants, but was a question about natural kinds. And this kind of came out of talking with a lot of people about the ideas we had and hearing consistently this kind of question about co-occurrence. So this concern that there's sort of two twin concerns. One was kind of, don't these things co-occur too highly for you to be able to parse them apart really? And second of all, how do you really know that those links are the ones that you're describing? Kind of a question about like, isn't this just a trick of your statistics? And and I didn't really think it was, but I also was a little worried, like, would this really emerge and out of the data if I used a more data driven approach and I didn't have such strong hypotheses as I was kind of setting things up. And so one of the reasons why people were concerned about this, and I was concerned about this, is that if you do a cluster analysis of just adversity exposure, if you do a factor analysis of exposure, if you do like a latent class of exposures, what you observe is that there are things that co-occur with poverty. There are things that there's institutionalization, which is an unusual exposure that also happens. And then there's things that co-occur with abuse. But you don't really see the thing that we would have expected, which is something like poverty, language exposure, neglect, and institutional rearing being associated with each other. And then in contrast, community violence, sexual abuse, domestic violence, and physical abuse, right? That's like not really a thing that you observe. And I wanted to think about a way to solve that problem. And so I came up with this idea of using network analysis, which is, increasingly familiar and common because of neuroscience work to me, as a way of kind of solving this clustering problem. And network analysis seems useful because it's a really general solution to examining the structure within a set of relationships. You don't really need data to be particularly similar to each other or to have any special properties in order to be able to do this. And it also has lots of measures for describing clustering and the role of specific nodes. And my idea here was that if you added to this pot of stuff the outcomes that we expected to be specifically related to our exposures, then you could sort of get this to organize in the way that we would have expected. So if you add cognitive control and cortical thinning and fear learning and emotional control and amygdala reactivity into the pot, maybe what you would get out is the kind of organization that we expected to find. And so then you'd still see these original pathways, but now there'd be all these other pathways sort of like pulling apart the networks and organizing them. I paired up with Bill Shee, who was at UNC and then went to Amazon because they had more money, to build the sort of approach that we used here. So this is not my methodological approach, this is his methodological approach, but we worked closely together on it. And first we wanted a way to sort of create an average network, but also to get a normal curve around that. And so what Bill thought would be useful to do is actually to use imputation. So we imputed 1,000 data sets, we made networks out of each of those data sets, and then we averaged those together to create an average network, and then also a confidence interval around that average network. And then we asked whether or not that average network was or wasn't similar to a cumulative risk model. So that would be there are sort of two different versions of the cumulative risk model. One would be all the exposures and outcomes are equally linked with each other. And one would be all exposures are linked with all outcomes, but they're not necessarily linked with each other. That's the one phase where you can ask whether or not this network is or isn't similar to a cumulative risk model, and kind of asking if it is or isn't similar to a DMAP model is the other question. So in that case, we took an original data set, imputed it, made a bunch of networks, and then we performed clustering on each of those networks to sort of get two different clusters of, or however many clusters it wanted to give us out of the data. We did some consensus clustering to say, here's what the clustering appears to be in these networks. And then we asked whether or not that consensus clustering was or wasn't similar to the proposed deprivation and threat model. We first did this in two different samples of teenagers. So one in which children reported only, and then one in which both children and parent reported on their findings. And the results were pretty similar. So here we have their performance on things like the emotional Stroop, their overall performance on like the emotional Stroop, and also their Stroop adaptation score, which is like the degree to which emotion is disrupting their performance. We have performance on matrix reasoning, arrow switching task, a simple inhibitory control task. And then we have things like and performance on vocabulary. And here we have parental education, which is really our only metric of deprivation. It's sort of a risk factor for deprivation. Or sorry, not our only metric, because we also had neglect, which importantly did not cluster with deprivation items. And then over here in what we are calling our threat cluster, we have sexual abuse, physical abuse, direct community violence, and domestic violence, and this emotional Stroop adaptation. Now, physical neglect clustered with threat. which is consistent with previous work showing that kind of like maltreatment cluster. So it was interesting to see that we did get 2 clusters. We could have gotten many, but we got 2. They do generally look like deprivation and threat, and they are, not statistically significantly different than the proposed DMAP structure. They're very statistically significantly different than the cumulative risk clustering model. So it does look like close to the DMAP finding, which was exciting. And we replicated it in a whole different data set, and it had some different people reporting on things and had slightly different measures and found exactly the same thing. So that was great. But we also, again, found that physical neglect clustered with threat, with the maltreatment variables. And in both of those studies, people are self-reporting, adults, either kids are self-reporting on their own experience or parents are self-reporting on their behavior. And in both of those cases, it's from the same exact questionnaire measure that we're getting our physical neglect and our physical abuse measures. And so there's a lot of measurement variance that is probably contributing to them clustering together. But I still proposed that they wouldn't, and they did. So there you go. So we finally were able to follow it up with a sort of a richer set of measurements, and we were excited, and also with a larger sample. So we took the same Fragile Families data set that we had used previously in the SEM model, and we tried to do this network analysis with it. I actually technically started this analysis before the other one, but it's done later. But we didn't actually know the outcomes of the SEM model when we started running this. So in our defense. So again, there's about 2,500 kids. This is a representative sample. This is not our data, obviously. This is Jeannie Brooks-Gunn is one of the PIs on it. It's in New York, and it's from data collected a while ago. The kids are older now. I think they're in their late teens. But it was a representative sample of urban centers with high rates of single-parent families. We're going to look here at the age 3 data. So that's just within that one age point. Our associations are controlled for child age, gender, socioeconomic status, parent race, parent cognitive function, and then because of network analysis, there's all the other variables in network analysis. And then we also controlled here for prenatal risk. So here we're controlling for a variable which indicates the child's prenatal exposure to alcohol, smoking, and illegal substances, their low birth weight, and whether or not they had prenatal visits. We have lots of measures. So there are some, I'm grouping them here in terms of like how we think of them. So we have things that we think are cognitive and social stimulation, which is a bunch of measures about books in the home and time you're spending with your kid and bedtime routines, and observed positive stimulation from the parent. We have material deprivation. So food insecurity, the parent's report of neglect, and the observation if the child appears neglected. We have some variables that we weren't really sure what would happen with them: frequent separation, eating meals together, difficulty with childcare. And then we have threat exposures. So this is spanking within the first year of life, and these are sort of community violence, domestic violence, and abuse variables. Our outcomes are pretty limited. We have the Peabody Picture Vocabulary Test, which is a vocabulary measure. And then we have symptoms of psychopathology and positive social behavior. So we again see the expected 2 cluster, we see 2 clusters as we expected. And I'll go through and tell you a little bit about those clusters. The cognitive stimulation clusters, they cluster with the Peabody Picture Vocabulary Test in one part of the sample, and the threat variables clustered together with symptoms of psychopathology and social skills in the other part of the sample, or the other part of the network graph. All the threat variables and all the chaos or unpredictability variables clustered together. So it's things like whether or not the parents are able to have dinner with their kids and things like that. They cluster together with the threat variables. And self-reported neglect, again, just like in the other 2 studies, did cluster with threat, but observed neglect, the way that the person who went to the home and saw the child, observed the child, actually does now cluster with deprivation. So the hypothesis that some of the sort of close association between neglect and abuse was due to measurement variance does seem like it's supported by this finding. We also then put socioeconomic status into the and race into the network structure in order to see whether or not those there's sort of 2 questions. One, are those going to reorganize the network to make it just one big clump of cumulative risk, which is kind of one of the ideas. And the other question was whether or not they were serving as important nodes in the network. And we found that they didn't just pull the network all together. So you do still see the deprivation and threat clusters separate from each other. They ended up actually being in the deprivation cluster, the education and race and income needs. But also importantly, they're kind of serving as really important informational nodes. So if you do random walk, for instance, they are a node through which lots of information travels to get through the network. So they're kind of these important informational nodes in the network. So we don't see a cumulative risk cluster. We do see the deprivation variables cluster with cognitive outcomes, threat variables cluster with emotion outcomes, with symptoms of psychopathology at least. And we do see the observed neglect now does cluster with deprivation. So that's sort of, I think in conclusion from the part of my talk, which is a bunch of data, I would say we've got this data that deprivation and threat differentially predict child development outcomes. And we've used both data-driven and a priori approaches. I can now take a minute and answer some questions I can tell you about the studies in my lab. I am going to take a minute to see if people are interested in asking questions. And if you're not, I'll tell you some more stuff. So this is your moment to stop me from talking.
My question was related to early childhood and behavioral inhibition, so I will hold off on that.
I thought that it's interesting the overlap between what you're seeing in terms of physical threat and neglect, and the cortical thinning that you're showing with the institutionalized children. Because I've always thought it was really interesting how, in terms of neural structures involved in, nociception, also overlap with, social pain and exclusion and things like that. Like a lot of the regions that you showed, such as the anterior cingulate, anterior insula, and it's interesting to see that kind of overlap. And I, wonder if it seems like all of these are kind of piecing together, including with your network analyses. And I wonder what you think about that, if these are sort of overlapping neural and behavioral sort of domains.
Yeah, it's interesting. I think you're sort of positing that maybe what I'm seeing in the deprivation findings is something that's kind of like a social pain, like kind of more consistent with the social pain kind of loneliness literature. Is that sort of what you were proposing?
I suppose. I mean, there's a lot of ways you could look at it, but there's this large degree of overlap between these different domains and neural structures.
Yeah.
And your own findings.
Yeah, that's interesting. I mean, I think that's super interesting. I don't really know very much about the social pain literature, so I had to go read that in order to see whether or not I thought that was like to observe the overlap that I think you're seeing, but I trust you that it's there. And so I think the way that I really think about the impact of deprivation is in a super cognitive way. So I really think of it as like the association cortex also requires input to develop normally, and the input that shapes the sensitive periods in association cortex is like this like cognitive stimulation and social stimulation that your parents do, but it's not like sort of, I think I've kind of like shied away from the part where that's distressing, I guess is what I'm saying. Like I've kind of been like, sure, but like, who cares what is actually going on for your brain? And I think that it's an interesting point. I mean, one of the things I think we're trying to grapple with now is how to think about that social piece of it. And so it seems possible that could be one link that we could make to kind of bring back in the emotional deficits that come along with a parent not having a parent or having a parent who's really underinvested.
Yeah, well, and another interesting thing about those same regions is that they also overlap with the salience network. So it's likely that it's not really the phenomenological experience of pain or social pain or exclusion or whatever, but something else, maybe more related to salience.
Yeah. Yes, they do. And I think also the findings, which I didn't show any of the imaging findings, but in terms of sort of the findings that we have relating exposure to abuse, controlling for deprivation, they're all basically in paradigms in which we're showing people like the emotion regulation task where you're showing somebody something bad and you're comparing it to showing them something boring, and what you just see like a whole salience network that's more active for kids who've been exposed to abuse. And I would say that that's like pretty consistent. It's like the amygdala, the insula, and the ACC, and I think that that's kind of consistently observed. And yeah, also a bunch of temporal cortex actually, but I think that one of the things that the salience network is sort of a place where these two things come together, because I think of them as like being these separate exposures, but it is true that like you see this kind of like negative valence, something bad is happening, activation really being related to threat, and then what I have like historically thought of as being really just sort of conflict detection kind of like activations related to deprivation. But the salience network is what ties those things together for sure. Zainab, what was your question about behavioral inhibition in early childhood?
So there's a lot of, I was interested by the first part where you were talking about the sensitive periods, and I was thinking about when that could be in humans. And one way to potentially investigate that behaviorally would be to look at stuff like Nathan Fox's behavioral inhibition stuff, right? Mm-hmm. And that could be an easy way to test things like novelty preference or explorative behaviors. And later on, we know that behavioral inhibition early on relates to things like anxiety and social anxiety later on in life. And so that would be one way to relate it to. I know you're not thinking about it in terms of like anxiety disorders and so on, but it might be a nice way to be able to tease apart like when the different sensitive periods are.
Yeah.
Or like fear avoidance and fear behavior and things like that in humans, and then tie that into the to your model.
Yeah. I think that would be, I think the questions around sensitive period in the model are sort of pressing and need to be sorted out. So I think that would be really interesting to do. And it would be, we do need to figure out whether or not it matters if this is really I talk about it as species expectant development. So as if there is a sensitive period and that your brain is requiring that to develop normally. That's the way that I've always talked about it and the way I kind of thought about it. But I think recently I've been kind of pressed to really say if that's true or not. And if it is true, if it's really like a sensitive period thing, or if it's more like just experience dependent development that can happen all along the course of development. And I don't know. Yeah, but I do think we should figure it out. I think that like the hints that I have that it's maybe more, I think are kind of in line with it being more experience dependent. So the thing that basically the kids who've been institutionalized, they were randomized in early childhood, but that randomization also sort of then resulted in a cascade of different things happening to them for the rest of their lives, right? So the kids in foster care have overall had better care for longer. So they're continuing to get more stimulation, and the kids in care as usual have overall had worse care for longer, even though there's like variability in there. That is generally what's happened. And for instance, there are things like in the Great Smoky Mountain study, they randomized kids to have more money, and they observe this improvement in their cognitive function. But that randomization happens when the kids are 9. Like it's super late. And so it's not like all of the action seems like it's really happening early. And I think I'm having a hard time figuring out how to marry the sort of conceptualization of things where I'm thinking of, like I was describing the theory by analogy, like I've been thinking about it as this like, you'd get different cortex because your brain was waiting for different inputs, and having to marry that conceptualization with like the data, which seems like it's more like an experience dependent development, I think, actually. But if you have ideas of how to use Nathan's subjects to do that, I would be interested in it, because I don't really know right now.
I think you could use his tasks rather than his subject.
Yeah.
You could collect, more and probably better data.
Yeah, that's interesting. Yeah.
Do any students have a question?
to start talking if you guys don't ask questions.
We can do a few minutes of this then.
All right, I'm going to tell you guys some stuff and then maybe you'll have ideas then. Okay, so I finally managed to get funding. It took a long time. It took much longer than I thought. It was a terrible experience. Yep. Here we are though, we got funding. So now I'm doing 2 studies, and both of them are designed to ask 2 different questions about the model. One of them is about asking, what does this look like in early childhood? So most of the literature, I reviewed one study in early childhood, but actually most of what we've got is in adolescence, where we're asking retrospectively about experiences. So here we're going in early when the kids are between the ages of 4 and 7, so they're 4, 5, or 6. We do a home interview and we get executive function at the home. We have them come into the lab and do an EEG task and a frustration task, which is sort of beside the point, but we have them do fear learning, and we measure IQ, again, some more executive function. We're getting some cortisol and immune function measures so that we can like talk a little bit about the links between the cumulative risk model and our model, and see whether or not there's some sort of some things we can think of as being more cumulative risk-like and some things we can think of being more DMAP-like. And then we have them come back and do an fMRI study where we're doing inhibitory control, emotion regulation, and reward processing. And I just want to tell you about the emotion regulation task. So this is like the only task I'm going to present, and well, in detail, and I'd be interested in hearing you guys' thoughts about it. So this is the old Oxner task that we've used in adolescence, where you tell people to do something like just look at an image or decrease your reaction to the image, and you sort of tell them how to do it. Then you show them a neutral, negative, or in our case, we show them a positive image sometimes. You ask them how they felt about it, and you tell them to relax. Here, we're trying to sort of make a version of this task that is more functional for little kids. And there was somebody who had done some of this work in little kids using EEG. And so what we did is we wanted to make it more like how little kids regulate emotions. So we gave them some scaffolding information. Then we tell them a story about what they're about to see. And then we show them a picture. And then we ask them, do they feel thumbs up or thumbs down about that picture? So the scaffolding story would be something like, now you're going to see some pictures of kids at the doctor. You won't get sick like these kids. And then we'd say, this girl's at the dentist. She feels funny, but it doesn't hurt. And then we show this picture, somebody who's ostensibly not being hurt. And then in contrast, we might show them something like, now you're going to see some pictures of kids who are hurt. This girl's in trouble and her teacher is mad at her. So we're sort of doing the look negative and the regulation version of the task. And there's this other part of the task that we've kind of introduced all through the study. And Zainab, I don't know if you were thinking of like, when you're talking about sensitive periods, if you're thinking of Nim Tottenham's work, but what we introduced here is some stuff about whether or not your mom is present, because that's actually the way that the fear learning stuff in rodents is done, is actually the presence of the dam. And so we're trying to kind of replicate those sorts of things here. So we sometimes have, this is my son, Ollie. Sometimes we have your mom telling the reappraisal scaffolding information. So I'm the one saying, these kids are sick, but you're not going to get sick like these kids. And then we have someone else doing the rest of this kind of story stuff. Sometimes we have somebody who's not your mom doing the scaffolding thing. And then the rest of the stuff is always by not your mom. And so we're able to contrast the 4 different conditions. Let me see if I have this. The 4 different conditions are whether or not it's a neutral. So all of this could be neutral, like it's just a picture of a lamp and this is a lamp and everything's neutral. It could be negative, like this kid is teacher's mad at them. It could be emotion regulation and your mom is scaffolding it, or it could be emotion regulation and not your mom is scaffolding it. So we have sort of 4 different conditions that we can compare among to get at this emotion regulation process. So that is one of the things, and that's one of the things we're doing. The second thing we're doing is a longitudinal follow-up, but it's 2. So do I need to end so that you guys can go about your businesses? And also, / does anybody have thoughts? Sorry.
Maybe like 1 minute or a wrap up, and And then we can,
So-
I didn't mean
A
stop.
woman oh, sorry. I'll just say the other thing that's basically like that's one of the new tasks that we're doing that I'm kind of developed for this, and I'm interested in people's thoughts on. The other thing we're doing is we have a longitudinal follow-up where there were 917 kids who were seen here when they were 2 to 5, and we got information about their anxiety, actually, Zainab, and also their exposure to adversity. And now we're bringing them back when they're like 15 years old, and we're going to do a lot of the same things that I just described to see how it interacts with anxiety across time. So
Very, very cool. Well,
oh,
hi, Margaret.
thank you.
Any questions from students or anyone else?
I'm going to do this really quickly though, guys, because I don't want to be a bad human. I want to show you my lab because they did all the work, obviously. Sorry, everybody. There they are. They're the best, the absolute best. I could not do anything without them. Okay.
Awesome. All right. Anyone have any thoughts or questions? All right. Well, thanks again, Margaret, for speaking here, and have a good weekend, everyone. I'm going to stop